Does every compound have a unique boiling point?
No, and this is one limitation of distillation as a separation technique. If two chemicals coincidentally have very similar boiling points, then it will be difficult to separate them using distillation.
What is precipitation?
In chemistry, precipitation means something different from when the weather forecaster on the news predicts rain. A precipitate is a solid substance that does not dissolve in a solution. And precipitation describes the process of the precipitate forming. This generally takes place when one or more soluble reactants in a solution undergo a reaction to form one or more insoluble products. The result is that a solid substance will form in a solution, either depositing on the bottom of the container or sometimes floating around in the solution. Depending on the circumstances, precipitation can be a desirable or undesirable result.
Why did Sir Robert Robinson, United Kingdom, win the Nobel Prize for Chemistry?
For his investigations on plant products of biological importance, especially the alkaloids. Robinson made significant advances in medicinal chemistry, both in synthesis and in gaining a better understanding of the mechanisms by which drugs work. His Nobel Prize was awarded for his work with alkaloids in particular, including quinine, cocaine, and atropine. Robinson studied and helped to understand how these types of molecules have their effects on the human body and mind.
What is Le Chatelier's principle?
Le Chatelier's principle tells us how to predict the effect a change in conditions will have on a chemical equilibrium. It tells us that a system at equilibrium will shift to counteract changes that disturb the equilibrium. These could be changes in concentrations of chemical species, temperature, pressure, or other conditions. The most commonly discussed changes involve changes in concentration of chemical species. So we'll just focus on those here. For this equilibrium, A and B, D, C and D, if we decrease the concentration of A, some C and D will react to replenish the A that is depleted, so the concentrations of C and D will decrease. As species A is replenished, more B will be created as well. So the net effect is that decreasing the concentration of A will also decrease the concentrations of C and D, and at the same time increase the concentration of B. More generally, decreasing the concentration of a reactant will cause the equilibrium to shift toward the reactants. Increasing the concentrations of other reactants and decreasing the concentrations of products. The converse is also true. decreasing the concentration of a product will cause the equilibrium to shift toward the products. Increasing the concentrations of other products and decreasing the concentrations of reactants. It is important to keep in mind that Le Chatelier's principle only applies to reversible chemical processes. Chemical equilibria. So everything we have said here does not apply to reactions that can only proceed in the forward direction.
What is ADP?
ATP, or adenosine triphosphate, is a molecule used as a source of energy in the body. The energy of an ATP molecule is stored in its chemical bonds, and it is released through hydrolysis of phosphate groups to form ADP, adenosine diphosphate. The ATP in our bodies is continuously recycled, and on average, each ATP molecule in our body will be used and regenerated over 1,000 times each day.
What is exothermic?
A process or reaction that releases heat.
What makes wine taste dry?
The dry taste certainly doesn't fall into one of the 5 basic groups, but it is known to be related to the presence of tannins.
How do you measure molecular weight of a polymer?
The most common way is based on size. The technique is known as size exclusion chromatography, or gel permeation chromatography. The sample is passed through a column that has a porous solid material. The smaller polymers can work their way into those pores, while larger molecules don't interact with the solid material. The biggest molecules, because they don't interact with the solid phase, come out of the column first, followed by smaller and smaller molecules. The time it takes for a polymer to get through the porous column is related to its molecular weight. Okay, technically it's based on the hydrodynamic volume, but let's let this approximation slide. In practice, these instruments are calibrated using standard polymer samples of a known molecular weight.
How many amino acids are found in humans?
There are 20 amino acids commonly found in humans. They are classified into 4 groups based on the identity of their side chain. The 4 groups are polar, nonpolar, acidic, and basic. These 20 amino acids are what make up all of the proteins and enzymes in your body. The interactions between the individual amino acids determine the overall conformation of the protein. Though the relationship between the sequence of amino Acids and the structure adopted by the protein is very difficult to predict.
What is vulcanization?
Natural rubber that comes directly from rubber trees looks nothing like your car or bike tires. It's sticky, doesn't hold its shape when it gets warm, and if you live where it snows, it can get brittle. That sounds like an awful material to make a tire out of. Vulcanization creates cross-links in the rubber with the addition of sulfur to the natural rubber chains and improves all of these properties.
How can I make black snake fireworks from items around my house?
Note, this experiment involves fire and flammable materials, so adult supervision is required. Also, check your local laws before attempting this experiment. Chemistry principles encountered in this experiment. Chemical reactions, combustion reactions, materials you'll need: sand, about 2 cups, lighter fluid, a small bottle of about 100 milliliters. Baking soda, 1 tablespoon, sugar, 4 tablespoons. Cup or bowl, an outdoor location where you can light the black snake firework safely without damaging anything. The procedure: in a cup or bowl, mix 4 tablespoons of sugar with 1 tablespoon of baking soda. Use the sand to form a pile, in your chosen safe outdoor location. And then create a depression in the middle of the sand. This depression is where you will ignite the black snake firework. Pour a small amount of lighter fluid onto the sand to wet it. Try this experiment first with a very small amount of lighter fluid, and, if necessary, repeat the experiment with incrementally larger amounts. It's better to start too small than too big. Pour your mixture of sugar and baking soda into the wetted depression in the sand. You don't have to use it all at once. Feel free to experiment with different quantities of the baking soda and sugar mixture. Carefully ignite the lighter fluid with a match and stand back. You should see the mixture create long snakes of black ash. The burning sugar and baking soda form sodium carbonate, water vapor, and carbon dioxide gas. The ash in the snake is composed of carbonate and burnt carbon.
How is coffee decaffeinated?
Consider the chemistry required to decaffeinate coffee. Most techniques use an extraction process to remove the caffeine from the green coffee beans before they are roasted. One method starts by steaming the green beans and then rinsing them with an organic solvent, usually dichloromethane, to pull the caffeine molecules out of the beans. The other method uses supercritical carbon dioxide to extract caffeine. The latter obviously avoids the use of toxic solvents, but is energy intensive. With all extraction techniques, it is challenging to only remove the caffeine and not the flavor compounds that we want to taste in our coffee.
How is inorganic chemistry relevant to biological chemistry?
Because metals are important to many biological processes. In the active sites of enzymes, see biochemistry, where the important chemical reactions take place. Metal centers are frequently crucial to the catalytic bond-forming and bond-breaking events. Metals are also important for maintaining gradients in ion concentrations, allowing muscles to move, and a variety of other biological processes. Bioinorganic chemistry is thus a huge field. And these two topics are often taught in conjunction with one another.
Why have chemists devoted so much focus to the chemistry of carbon?
Because it's everywhere. Carbon is the fourth most abundant element in the universe, 15th on our planet. And the building blocks of life, DNA, amino acids, all contain many carbon atoms. Many biologically active molecules and medicines rely on carbon to define their overall shape.
Why did Robert F. Curl Jr., United States, Sir Harold W. Crotto, United Kingdom, and Richard E. Smalley, United States, win the Nobel Prize for Chemistry?
For their discovery of fullerenes. This Nobel Prize was awarded for the discovery of a new form of elemental carbon in which the atoms are arranged in a closed shell to form a ball. These three scientists worked together to make this discovery. And the resulting new form of carbon was known as a fullerene. Fullerenes are generated when carbon vapor, obtained by intense pulsed laser irradiation, condenses in the presence of an inert gas. Fullerenes of a variety of sizes are now known.
What is an aliquot?
A sample taken from a larger amount of a material.
What elements are in your body?
Six elements make up all but 1% of the human body by mass. In decreasing order, they are oxygen, carbon, hydrogen, nitrogen, calcium, and phosphorus. Oxygen and hydrogen are so prevalent because of most of our cells are over 50% water.
What is styrofoam, R?
Styrofoam, R, is a brand name, owned by the Dow Chemical Company, for expanded polystyrene foam. It's 98% air, which is why those Styrofoam, R, coffee cups are so light, and actually buoyant. In addition to disposable food containers, polystyrene foam is used in building and pipe insulation. Packing peanuts, and that green stuff they use for holding together fake flower arrangements.
Why did Melvin Calvin, United States, win the Nobel Prize for Chemistry?
For his research on the carbon dioxide assimilation in plants. Calvin received the prize for his work on what is today known as the Calvin cycle, which is the process by which green plants affect the fixation of carbon dioxide. Or in other words, the incorporation of carbon dioxide molecules from the atmosphere into other molecules. Calvin identified that there is a close connection between the metabolism of carbohydrates and photosynthesis. This process is quite complex, involving 10 different intermediates and 11 different enzymes to catalyze each step of the reaction.
What is a quantum?
A discrete amount of energy, electric charge, or another physical quantity.
How is a band gap relevant to the design of photovoltaic materials?
The band gap of a material determines what wavelengths of light a photovoltaic or any other material can absorb. Photovoltaic materials need to be capable of absorbing the wavelengths of light that the sun emits. And this requires that they have a band gap that spans the appropriate range of energies.
Why did James Batchelor Sumner, United States, win the Nobel Prize for Chemistry?
For his discovery that enzymes can be crystallized, and John Howard Northrop, United States. And Wendell Meredith Stanley, United States, for their preparation of enzymes and virus proteins in a pure form. Sumner was the first to provide concrete evidence that enzymes and proteins could be crystallized, paving the way for further research in this area. Northrop and his co-workers explored the conditions that led to formation of crystalline proteins, and mastered this art, pioneering the way for modern scientists working to crystallize proteins and viruses. Stanley demonstrated that viruses can be crystallized in the same way as proteins, and then showed that viruses are, in fact, proteins themselves.
What is osmometry?
The process/study of measuring the osmotic strength of a solution.
What is an organometallic complex?
These are molecules with bonds between carbon and a metal. This includes alkali and alkali earth metals, groups 1 and 2 of the periodic table, the transition metals. Groups 3 to 12, including the F block, and sometimes group 13 metals are also included. The bonds between metal and carbon can vary widely in their ionic or covalent character. The bonding between a metal and carbon is mostly ionic in two situations. 1. If the metal is very electropositive, as in groups 1 and 2, or 2. If the carbon group is a stable anion. By being delocalized via resonance as in cyclopentadienyl anion. There are also organometallic molecules with more covalent bonds between the metal atom and the carbon atom. This is usually seen with the transition metals or elements like aluminum. The nature of the metal-carbon bond plays an important role in how organometallic complexes react.
Is ceviche really cooked by lime juice?
The results of cooking with heat and with acid are similar, but of course they are not exactly the same. Both heat and acid serve to denature proteins in food, which is a technical way of saying that the molecules change shape. With access to any number of shapes that it couldn't exist in beforehand. The molecule finds new ways to react both with itself and with other protein molecules. These recently liberated proteins quickly form a solid network, which is why fish gets firmer and whiter when you add lime juice. And it's the same reason that egg whites turn opaque and get harder upon cooking.
What is an anion?
A negatively charged ion.
What is nuclear fission?
Nuclear fission is essentially the opposite of nuclear fusion. Here, a single nucleus divides into two smaller nuclei. In the case of heavy atoms, this is often accompanied by the release of heat. For example, the radioactive decay of uranium-235 can be used to generate the heat used to drive turbines to generate electricity in nuclear power plants. The use of nuclear fission to harness energy for use by humans is typically considered the much more viable choice, as opposed to nuclear fusion.
What is rayon?
You probably know what rayon fabric looks or feels like. The best Hawaiian shirts and much of 1980s fashion were made of it. What's fascinating about rayon is that it is not really a synthetic or a natural fiber. Rayon is a chemically modified cellulose polymer, first prepared in the 1850s. While there have been a number of ways of preparing this artificial silk, the viscose method led to the first commercial production of rayon. This method treated cellulose with a combination of sodium hydroxide and carbon disulfide as indicated below.
What is materials science?
Materials science is a field at the intersection of the basic sciences and engineering with a focus on the relationship between the microscopic, atomic or molecular, structure of a material and its macroscopic properties. Many techniques relevant to chemistry are used to characterize materials, and the descriptions of the underlying microscopic structure of the material are discussed with regard to solid-state chemistry. Thus many components of materials science represent applications of chemistry. In this section we will provide a brief introduction to materials science, with a focus on some of the topics that are more relevant to chemistry.
What is a microwave?
Electromagnetic radiation with wavelengths of 1 mm to 1 meter. Just longer than infrared radiation, but not as long as radio waves.
What is the Grignard reaction?
The Grignard reaction is one of the most well-known and powerful reactions in organometallic synthesis, primarily due to its ability to readily form new carbon-carbon bonds. In this reaction, a Grignard reagent reacts by adding to a carbonyl functional group of an aldehyde or ketone and forms a new carbon-carbon bond at the carbonyl carbon. Grignard reagents, which are organometallic species that carry out the Grignard reaction, are typically formed by adding magnesium metal to an alkyl or aryl halide, R1Br in the scheme below. The discovery of this important reaction was awarded with a Nobel Prize in 1912, and the reaction is actually also named after the French chemist François-Auguste Victor Grignard, who discovered it.
What is a levorotatory?
Having the property of rotating plane polarized light counterclockwise.
What is distillation?
A purification technique that separates substances based on their differences in boiling points.
What is a neutron?
Neutrons are the other principal component of the nucleus of an atom, along with protons. The neutron is neutral in charge and has a mass roughly similar to that of a proton. Atoms of the same element that contain different numbers of neutrons will generally still have the same behavior as one another in terms of chemical reactivity properties. Both protons and neutrons are, in fact, made up of even smaller particles. But chemistry doesn't usually deal with these even smaller bits.
How do solar cells work?
The sun gives off 1000 watts of energy per square meter of the Earth's surface, which, if we could harness all of it, is more than enough to fulfill all of the world's current electricity needs. It's not easy to do this, however, which is why we still rely primarily on other energy sources. A solar cell, or photovoltaic cell, harnesses energy from photons of sunlight by having them excite electrons in a material. An electric field is established within the solar cell using a process called doping. And this makes it so that the excited electrons can only flow in one direction. Thus, when sunlight strikes the solar cell, a current flows. And this is the basic principle on which solar cells operate to capture energy. The harnessed energy can either be used immediately or it can be stored for later use. Designing more efficient solar cells is a very active area of research. And you can expect their efficiency to continue to increase in coming years.
What is a compound?
A substance composed of more than one element.
How can I make slime?
Chemistry principles encountered in this experiment. Hydrogen bonding synthesis polymer chemistry cross-linking materials you'll need, water Elmer's glue, about 4 ounces. Or 120 ml, borax powder, 4 to 5 tablespoons, bowl measuring cup small jar. Doesn't need to have a cover, spoon or stirring device. Food coloring, optional. The procedure. Pour about 4 ounces of white glue into the jar. The glue contains several components, including the polymers polyvinyl acetate and polyvinyl alcohol. See polymer chemistry. Polyvinyl acetate contains oxygen atoms that can serve as hydrogen bond acceptors. And polyvinyl alcohol contains hydroxyl groups that can serve as either hydrogen bond donors or acceptors. Add 1/2 cup, 4 ounces or 120 ml, of water, and stir it in until it mixes with the glue. Note that there is already some water in the glue, and we are just adding more. Optional, add food coloring to the mixture to give your slime some color. In the bowl, mix 1 cup, 8 ounces or 240 ml, of water with 4 or 5 tablespoons of the borax powder. Stir this well. Here we are preparing a solution of borax so that we can add it to the mixture of glue and water in a more uniform fashion. While mixing, slowly add the glue and water mixture to the solution of borax. As you mix, you should observe the slime forming. Pick it up with your hands and knead it until it seems fairly dry. There will still be extra water left behind in the bowl, that's nothing to worry about. The borax serves to cross-link the polymers by forming hydrogen bonds with the oxygen atoms of the polymers in the glue. These interactions can readily rearrange to form new hydrogen bond donor-acceptor pairs with different oxygen and hydrogen atoms, which is what makes the glue so stretchy and readily deformed. When you are done with it, you can store the slime in a plastic bag in the refrigerator.
What is the Coulomb's law?
Law describing the force between a pair of charged particles separated by a distance.
How do your muscles work?
Muscles allow you to exercise and move heavy objects and are necessary for basic activities like breathing, pumping blood, and pretty much anything else you do. On a molecular level, muscles work based on the binding, movement, and rebinding of molecules called actin and myosin. This process involves the hydrolysis of ATP to generate energy. For your muscles to move, myosin first attaches to actin, forming a bridge. At this point, ATP and a phosphate group are attached to the myosin. The myosin bends, this is what actually controls movement, releasing the ATP and phosphate. Then a new ATP molecule binds again, and then the myosin releases the actin. The ATP is then hydrolyzed, putting the myosin back into its original position. At which point the cycle can begin again.
What are the lanthanides and actinides?
The actinides and lanthanides make up the F block of the periodic table. These two rows are frequently separated from the main table. But that's just so the periodic table isn't so wide on a piece of paper, seriously. Many of these elements have radioactive isotopes, but the rate at which they decay can vary immensely. For example, 238U has a half-life of 4.5 billion years. But 234Pa has a half-life of only 72 seconds.
What is potential energy?
Potential energy describes all of the non-kinetic energy associated with an object. This energy can be the energy stored in chemical bonds, in a compressed spring, or in a variety of other ways. Another example is gravitational potential energy. like that associated with a ball sitting at the top of a hill. Since there are many types of potential energy, there isn't a single equation that describes them all. Since the value we assign to potential energy is always inherently described relative to some choice of a reference value, we can only actually measure changes in potential energy in a meaningful way. A closed system can exchange potential energy for kinetic and vice versa. But the total energy must always remain constant. This is stated in the first law of thermodynamics, which we'll get to soon.
What is a protein?
Proteins consist of one or more long chains of amino acids that fold up into a specific arrangement, and each is presumably capable of some kind of biological function. Though we don't know for sure that every protein has a biological function, the function of each protein is one thing many biochemists are still working hard to figure out. Some proteins act to protect an organism from invading pathogens. Some help to carry messages between different areas of the body. Some are responsible for movement of muscles, and some provide structural support for cells. Many proteins also fall into a class called enzymes, which is a name for proteins that catalyze specific chemical reactions in biological systems.
What is a metalloid?
An element, compound, or alloy that has metallic and nonmetallic properties.
Why do NMR instruments use big magnets?
The purpose of using big magnets is, not surprisingly, to establish a strong, uniform magnetic field in the spectrometer. This creates an energy difference for the nuclear spins that are aligned either with or against the magnetic field. It is this energy difference that is being measured in an NMR spectrum, which can be related to molecular properties like the electron density surrounding a given nucleus.
What is carbocation?
A cation in which a carbon atom bears a significant fraction of the positive charge.
What is tribology?
Tribology is a subfield of materials science dedicated to studying the wear of materials. This may include the effects of friction on a material as well as how to better engineer surfaces or lubricate interfaces between surfaces to extend their lifetime.
Why is arsenic so poisonous?
Arsenic interrupts some of your body's most basic and common biochemical pathways. Arsenic, particularly as 3+ and as 5+ oxides, interferes with the citric acid cycle and respiration. Specifically reduction of NAD and ATP synthesis biochemistry. If that weren't enough to kill you, and it probably is. Arsenic also boosts the level of hydrogen peroxide in your body, which causes another whole set of problems. Unfortunately, these toxic forms of arsenic are not only water-soluble but can be found in well water from natural sources and man-made contamination from mining.
How can I observe layers of immiscible liquids?
Chemistry principles encountered in this experiment: density, miscibility, polarity, materials you'll need asterisk. Honey, pancake syrup, liquid, dish soap, water, vegetable or cooking oil, rubbing. Alcohol, lamp oil, a tall glass of water or other container, optional, food coloring to improve visibility asterisk note. For this experiment, it is not necessary to have every material listed. The procedure: pour the densest liquid into the glass first. Note that the liquids above are listed from most dense to least dense. Try to avoid letting the liquid run down the sides of the glass. Gently pour the second liquid on top of the first. One way to pour it a little more slowly is to pour the liquid over another object such as a butter knife or the backside of a spoon. Allow each layer of liquid to settle for at least a few seconds before adding the next liquid. You'll notice that, instead of mixing, the liquids tend to stay in separate layers. The reason for this is that they are immiscible, which means that it is more thermodynamically favorable for the liquids to stay separated in layers and to form an interface than it is for them to mix together. Whether or not two liquids will be miscible is dictated by the details of the entropic and enthalpic factors associated with the mixing or separation of the two liquids in question. This is often directly related to whether or not the compounds have similar polarity. For example, we know water is a very polar substance, while vegetable oil is composed of primarily long, nonpolar hydrocarbon chains. These do not interact favorably with one another, and prefer to stay in separate layers. Continue pouring the third, fourth, etc. liquids on top of each other in order of decreasing density. You can just follow order of your liquids in the list above. As you pour in the successive liquids, they should continue to form separate layers. The densest liquids are affected the most by gravity, so these tend to stay below the less dense liquids. These liquids are not miscible, so they do not mix together to form a single solution. In truth, if you wait long enough, some of these liquids will mix together. But it will take a while. That's it. You should now see a series of separate liquid layers in your container.
Why does the sun glow?
The fusion processes happening near the core of the Sun cause energy to be given off in the form of photons. See Physical and Theoretical Chemistry. The photons given off in the core of the Sun collide with other atoms, which absorb photons and, in turn, give off additional photons. This process repeats, potentially millions of times, before photons at the surface of the Sun are emitted off into space. As a side note, everything tends to emit radiation in this way. At least to an extent. It's just that most things on Earth are not nearly as hot as the Sun. Even your own body releases electromagnetic radiation. But the photons coming from your body are in the infrared region of the spectrum, so we cannot see them with our eyes. However, infrared cameras can use the photons given off by a person's body to locate people or animals, in this way.
Does all the alcohol really boil off when I cook with wine?
Not really, no. It's common lore that when you add red wine to pasta sauce that the alcohol evaporates rather quickly. This idea is supported by the fact that alcohol has a lower boiling point than water, so it should evaporate quickly. People who actually studied this, however, have shown that even after an hour, 25% of the alcohol you added is still in the sauce. If you want a truly non-alcoholic marinara, you need to simmer for at least 2.5 hours.
What molecular properties lead to higher boiling points?
There are a few factors that play important roles in boiling points of substances. The first is molecular weight. In general, heavier molecules have higher boiling points, which is pretty straightforward considering how we defined boiling point above. Heavier molecules take more energy to move from the liquid to the gas phase. All other properties that affect boiling point deal with intermolecular forces, or interactions between molecules. Think of this like the affinity of one molecule of a substance to be attracted to, or stick to, another molecule. Non-covalent bonds, like ionic or hydrogen bonds, significantly increase boiling point. Why? Because to move to the vapor phase the molecules typically have to break these interactions. Dipole interactions and van der Waals forces have similar effects, see atoms and molecules. But these interactions are weaker, so the effect they have on boiling points is smaller. Finally, branching of the carbon backbone of a molecule is also frequently touted as a factor leading to lower boiling points. While this is true, it is really the weakening of van der Waals forces at work here too.
How are elements named?
The names of elements often have interesting origins. They have been named after people, places, colors, mythological creatures, or for a variety of other reasons. Some are named after scientists, such as curium, after Marie and Pierre Curie. Lawrencium, Ernest Lawrence, Seaborgium, Glenn Seaborg, Mendelevium. Dmitri Mendeleev, Einsteinium, Albert Einstein, and barium, Niels Bohr. Others are named after places, such as Lutetium. Lutetia means Paris in Greek, Californium. Berkelium, Berkeley, California, Americium, Dubnium, Dubna, Russia, Hassium, Hessen, Germany. Yttrium, Ytterbium, Terbium, and Erbium, these last four being named after Ytterby, Sweden. Tantalum, Tantalus, Niobium, Niobe, Promethium, Prometheus, Uranium, Uranus, Neptunium, Neptune. Plutonium, Pluto, Palladium, Pallas, and Cerium, Ceres, are all named after mythological creatures. Though elements can take on different names in different countries, the commonly accepted names are those agreed upon and assigned by the International Union of Pure and Applied Chemistry, IUPAC.
What is amorphous?
A solid without a repeating, ordered structure.
If polar bears have black skin and clear fur, why do they look white?
Polar bears appear white for the same reason that a pile of snow looks white: reflection. If there are a lot of surfaces that reflect light, either in the polar bear's fur or a snowdrift, then any light that hits the object will be bounced around many times before coming back toward your eye. Most wavelengths of light are scattered equally well, so you end up with the object looking white. In other words, no wavelengths are absorbed, which would give rise to a color.
What is the Le Chatelier's principle?
States that any change, concentration, pressure, temperature, volume, to a chemical system at equilibrium will cause the equilibrium to shift in order to counteract that change.
What is a nucleotide?
Nucleotides are one of the basic units for another class of biomolecules, the ones that join together to form your DNA and RNA. The long names for these are deoxyribonucleic acid and ribonucleic acid. DNA and RNA are the macromolecules that store the genetic information in your body. A nucleotide consists of three parts: a nitrogenous base, a sugar, and a phosphate group. The identity of the nitrogenous base determines which letter in the genetic code is associated with the nucleotide. while the type of sugar determines whether the nucleotide is a ribonucleotide or a deoxyribonucleotide. Which essentially just tells us whether it's going to be a part of RNA or a part of DNA.
Why do hard-boiled eggs spin, while raw eggs don't?
Hard-boiled eggs are solid all the way through, so when you spin the egg, all the energy you apply goes into spinning the whole object. Raw eggs have yolks that are free to move about the interior of the egg, however. So when you spin a raw egg, the yolk moves to the outer edge of the inside of the egg, which consumes some of the energy you applied. The other difference you can see is what happens after you stop these two eggs from spinning. Stopping the hard-boiling egg stops the entire system. Because the yolk is trapped in the solid, cooked egg white. Inside the raw egg, however, even after you stop it, the yolk is still spinning around. So if you take your finger off of a raw egg that was just spinning, it can start moving again, seemingly on its own.
Is it good to drink chocolate milk after a workout?
Yes. Milk contains a significant amount of protein, roughly 8 to 11 grams per cup. It is often recommended to consume 15 to 25 grams of protein after a workout session. So drinking 2 to 3 cups of chocolate milk would meet this suggested quantity. In comparison to regular milk, chocolate milk has about twice as many carbohydrates, which is good for soothing sore and tired muscles. Of course, it also serves to rehydrate your body from the water lost during the workout. Moreover, milk provides you with nutrients like vitamin D and calcium.
What is the reduction potential of an electrochemical cell?
The reduction potential of a species describes its tendency to accept electrons. Or, in other words, to be reduced. A full electrochemical cell typically involves two separate half reactions, each of which is associated with its own reduction potential. The overall reduction potential for an electrochemical cell is the difference of the reduction potentials for the two half reactions.
What is fusion and how might it be used as an energy source?
We looked at fusion in nuclear chemistry. To recap, fusion involves the combination of two nuclei to form a single nucleus. For lightweight nuclei, this process typically involves the release of energy. Unfortunately, it's currently very difficult to make fusion happen under practical conditions. Nuclei typically have to be accelerated to very, very high speeds to make fusion take place.
What is an isotope?
Isotopes are atoms with the same number of protons and electrons, but with different numbers of neutrons. Since the numbers of protons and electrons effectively determine the reactivity of an atom, isotopes have the same basic chemical properties and are the same element. They have different masses, though, since they have different numbers of neutrons. The various isotopes of an element are usually present in relatively fixed ratios throughout nature. But in some cases the ratio can depend on the environment or molecule in which they are found. For example, the element carbon most commonly exists with 6 protons, 6 neutrons, and 6 electrons, referred to as carbon-12, the total number of particles in the nucleus. A small fraction, however, have 6 protons, 7 neutrons, and 6 electrons, carbon-13. Roughly 99% of carbon atoms have 6 neutrons, but most of the remaining 1% have 7 neutrons.
What is an allotrope?
Different arrangements of atoms of a single element, e.g., diamond and graphite are allotropes of carbon.
Is it true fresh eggs will sink in water, while bad ones will float? Why?
Yes. As an egg goes bad, proteins and other chemicals decompose and release volatile molecules like carbon dioxide, CO2. The shell of an egg is somewhat porous, so these gases can escape, decreasing the mass of the egg. Since the size of the egg doesn't change, assuming you don't break it, the density decreases as the egg ages, and it eventually becomes less dense than water. Whether it becomes buoyant exactly when it is spoiled or weeks after, we can't say. That's why your nose is so good at smelling that awful smell so that you don't eat rotten eggs.
What are the basic units of length?
Almost every length scale used in science is based on the meter. Chemistry frequently deals with very small lengths, so while you're probably familiar with millimeters, 10 to 3 m, it's hard to have an intuitive sense about just how small a nanometer, 10 to 9 m, is. There's another length scale commonly used when talking about chemical bonds, the angstrom. An angstrom, A, is 1/10 billionth of a meter, 10 to 10 m. The lengths of chemical bonds vary depending on the elements and other factors, but are usually around 1 to 2 a.
What is a solvent?
The major component of a solution.
What is a free energy diagram for a chemical reaction?
A free energy diagram is probably easiest to understand by taking a look at one. See diagram, as we explain the key features. The y-axis measures the relative free energy of the chemical species we're dealing with, while the x-axis describes the reaction coordinate. It's common that going left to right is forward progress in the reaction. But this isn't necessarily the case 100% of the time. On the left we have our reactants. In general there may be any number of reactants, and here we've just denoted two species, A and B. The hill in the middle is the energetic barrier to the chemical reaction. And the quantity EA denotes the height of this energy barrier. The quantity EA is commonly referred to as the activation energy for the reaction. On the right-hand side of the diagram we have our products. Again, there can be any number of products, and here we've denoted them C and D. Finally we have the quantity G, which describes the change in Gibbs free energy associated with the reaction. The fact that the reactants are higher in free energy than the products tells us that this particular example is a spontaneous reaction. If the reactants were lower in free energy than the products, the reaction would not be spontaneous.
What is smog?
The word smog was originally derived from a combination of the words smoke and fog. And today it describes air pollution that, once in the atmosphere, reacts with sunlight to form additional pollutants. The main sources of this pollution are commonly motor vehicles, factories, power plants, and burning of fuels. The main chemical components of smog are ozone, nitric oxide, nitric dioxide, and volatile organic compounds. Smog is present in pretty much every large city around the world. It can be hazardous to human health and has been blamed for health problems and even deaths.
What is an atomic orbital?
Atomic orbitals are mathematical or pictorial descriptions of the locations of electrons in an atom. Electrons are tricky particles to understand because their location isn't easy to define. They can be thought of as clouds of negative charge surrounding a nucleus. And atomic orbitals describe the shapes of these clouds. Atomic orbitals can take on different shapes and sizes, but are essentially very similar from one element to the next. The number and type of orbitals that contain electrons play a central role in determining the properties of that atom.
What is electronegativity?
A measure describing how strongly atoms attract electrons.
What are MRI contrast agents and what are they used for in medicine?
Magnetic resonance imaging, MRI, see the modern chemical lab. Contrast agents are used to make tissues and other structures inside the body easier to view in an MRI scan. Many of these contrast agents are based on gadolinium with various ligands attached. The contrast agents are injected or ingested into the body, and they function by changing the relaxation time of the atoms observed in an MRI scan. The overall result is that these complexes improve the ability of MRI to see what is going on in your body.
What is a biosignature?
A biosignature is a chemical signature that can be observed from a distance that signals the presence of living organisms. These could include complex chemical structures associated with life forms or accumulated quantities of biomass or waste.
What is systematic error?
Systematic errors are unlike random errors in that they change the observed value from the true value in a consistent direction. One example would be if you read a thermometer from the wrong angle. You might consistently read the level of the liquid inside to be a few degrees higher than it actually reads. This would systematically bias the observed value toward a higher temperature than the true value. Another example would be if a scale were calibrated incorrectly, such that it read 5 grams even when no weight was placed on it. This could result in an observed mass that is systematically higher than the actual mass.
What is a cross-linked polymer?
When a bond is formed between two polymer chains, and technically not at the chain ends, the product is called a cross-linked polymer. Creating linkages between chains usually increases their viscosity, so more like molasses than olive oil, and creates elastic properties like those found in rubber bands. At higher levels of cross-linking, polymers can even become stiff or glassy.
What separates ancient and modern chemistry?
While there's not a clear, punctuating distinction between ancient and modern chemistry, there are a few major differences that separate the two. Modern chemists describe the world in terms of atoms, molecules, and electrons and have a relatively complete understanding of the basic particles that make up matter, at least insofar as is necessary to describe chemical transformations. Ancient chemists didn't have this information and relied less on experimental evidence and more on theory and mythology. For example, ancient chemists sought the philosopher's stone, see below, for which there was no verifiable evidence. But they were attracted to it for its mythological power to preserve youth.
What did Empedocles believe were the four basic elements?
A Greek named Empedocles, who was not from Miletus, but rather Sicily, was the first to propose the four basic elements. These four elements were earth, air, water, and fire. These elements had a much different definition from that which chemists use today, which we'll get to later. Unlike the modern definition of an element, Empedocles' understanding of an element did not require it to be a pure substance. Water, for example, was obviously not the only liquid Empedocles had ever encountered. Earth represented solids, water represented liquids, air represented gases, and fire represented heat.
How can I make glue from milk?
Chemistry principles encountered in this experiment: solubility, precipitation, filtration. Materials you'll need: hot water, from the tap, is fine, room temperature water 1 tablespoon. Of vinegar 1/2 teaspoon. Baking soda. Coffee filter cup, spoon, small bowl. Tablespoons powdered milk. The procedure: dissolve 2 tablespoons of powdered milk in 1/4 cup of hot tap water. Mix in 1 tablespoon of vinegar and stir the solution well. Recall that household vinegar is a dilute aqueous solution of acetic acid. At this point the milk should begin to form little blobs of insoluble material, curd. These are a substance called casein, which are a class of proteins found in milk. Proteins are typically soluble in water and aqueous environments. But the acetic acid from the vinegar causes the casein to no longer be soluble in this solution. Position a coffee filter on top of a cup and pour the solution through the filter to collect the curd. To further dry the curd, try to use the filter to squeeze out any remaining liquid. Dispose of the liquid in the cup, dry the inside of the cup, and then transfer the curd from the filter into the now dry cup. Break the curd into smaller chunks using a spoon. This will help it to mix more readily with the ingredients you will add next. Add 1 teaspoon of hot water, and 1/4 of a teaspoon of baking soda to the cup containing the chopped up curd. The mixture may foam a little from the reaction of the baking soda with the remaining vinegar, producing carbon dioxide gas. Stir this mixture, which should eventually become smooth and liquid-like. You may need to add a little more water or baking soda to reach a smooth, even consistency. Now you have your glue. You can use it just like you would any other glue, though you should test it out first to make sure it's working well before you use it for that science fair project you're finishing up.